Hello, I'm Dylan,
I'm Joe.
And I'm Maya.
and we are here showcasing our radio frequency biosensor for glucose water solutions. We were sponsored by Dr. Syed of the Electrical and Computer Engineering Department for a research-based project to create a glucose concentration measuring device operating in the radio frequency range. This project is heavily motivated by the prevalence of diabetes and the lack of a non-invasive glucose monitoring and testing method. The device was to be able to measure the glucose concentration by measuring the shift in the scattering parameters of a system measured by a VNA due to the change in the permittivity of the solution. First, the design started in ANSYS HFSS, where a triangular patch antenna with parasitic strips were designed to resonate between the frequencies of 1 GHz and 2.5 GHz. A triangular patch antenna was chosen to be the radiator due to its small footprint and ease of manufacturability. HFSS optimization methods were used to impedance match the antenna and optimize the gain and directivity.
Once we had a viable design, our prototype consisted of a CNC engraved copper-clad FR4 board. We attached a 3D printed cavity to contain our samples and soldered an SMA port to interface with the VNA. For data collection, we created a range of glucose concentration samples from 500 to 50 milligram per deciliter, which is the typical range of human blood. We also had a water sample for a control. Multiple trials were run on each sample where the VNA was used to run a frequency sweep. The frequency responses were recorded and post-processing was done using Excel and Python.
After collecting the data into the floppy disk, we processed it using Python. We then used Excel to plot the frequency response and attempted to correlate the resonant frequency with the varying concentration of glucose. We found potential for a correlation, but further research is required to confirm a correlation between the change of glucose concentration and resonant frequency. Future research should account for changes in temperature, external noise, and sensitivity antenna. Creating a resource of data would allow for machine learning algorithms to better predict the concentration of glucose being measured. The results of this project yield evidence that an RF biosensor is possible and should be further pursued.